in association with the Microbiologists Society of India. I'm Pratishtha, your host for the day. Before beginning the session, some general instructions. All the listeners are requested to keep their microphone and videos off throughout the session, as it would help us to run the session smoothly. There will be a Q&A round at the end of the session, but you're welcome to post your queries in the chat box at any time during the session. The concept of probiotics, which literally means for life, has emerged as a scientifically proven way to increase friendly bacteria that are absolutely essential for life and to improve gut health and immunity. Our gut microbiota is being identified more and more by scientists as a catalyst for strong physical and mental well-being. It is in this context that we have with us today Professor Bruno Pot, who is a highly renowned expert in the field of food microbiology and probiotics. He's going to share his thoughts on how probiotics and prebiotics play a significant role in our health and wellness. Welcome, Professor Bruno Pot. We are extremely grateful to you for accepting our invitation to today's event. Before we move ahead, please allow me to express my sincere gratitude to Professor Geeta Trilok Kumar, the Director of the Institute of Home Economics. Professor Trilok has always motivated students and faculty alike and has been a source of inspiration for all of us. We are also fortunate to have Professor Arvind Deshmukh, founder and the President, All India Microbiologists Society of India, with us today. Professor Deshmukh, we are thankful for your continued support and encouragement. I would request Professor Deshmukh to kindly share with us a few words. Professor Deshmukh.
Thank you, madam. Today's afternoon is an academic afternoon. The great speaker, Dr. Bruno Pot, is going to talk on a very, very important topic: prebiotics and probiotics. Literally, whole world or scientists from whole world are talking about prebiotic, probiotic and postbiotic. So it is a very important part of our life. And I'm very happy today Dr. Bruno will focus the light on prebiotic and probiotic. And today 100 participants are there, capacity is full. It means that what a good response your unit is getting for this lecture. Moreover, this lecture is being recorded. It will be available to all other scientists throughout the world, because we are going to display, we are going to circulate in all groups afterward. So, of course, credit goes to Dr. Sunita Agrawal ma'am and her team, all active teachers which are involved in her team. The collective efforts is today's result. So, as a president of Microbiologists Society, I appreciate Delhi chapter of Microbiology Society and all teachers taking efforts for the upliftment of the subject, for educating the students from your area. Thank you, madam. Thank you very much for giving me an opportunity.
Thanks, Professor Deshmukh, for the warm welcome. The president of the Delhi unit of MSI, Dr. A. Archana, is also with us. We really appreciate you taking out time for us from your busy schedule. I request Dr. Archana to kindly share with us a few words. Dr. Archana. Maybe there seems to be some network issue with Dr. Archana. Moving on, Dr. Sunita Agrawal, Program Coordinator, Star College Scheme, and State Coordinator, MSI, is also with us. She has been a source of invaluable guidance and counsel. It is because of her support that today's webinar is being conducted. Now, without further ado, I would like to invite Dr. Sunita Agrawal to welcome and introduce Professor Bruno Pot. Dr. Sunita Agrawal.
Thanks, Dr. Pratishtha. Good afternoon, everyone, and welcome to today's webinar on prebiotics and probiotics. It's my privilege and great honor to introduce Professor Bruno Pot to August gathering. Professor Bruno Pot started his academic career in 1983 as a microbial taxonomist at the Laboratory of Microbiology in Ghent, Belgium, where he obtained a PhD degree in microbiology. He then took part in many EU research projects on lactic acid bacteria and developed a particular interest in bioinformatics. After 14 years in research, he became scientist manager, Benelux and science manager Europe for the company Yakult Europe in the Netherlands. In 2001, he joined Applied Maths, a young Belgian bioinformatics company, taking up a part-time position as Director of Business Development. In parallel, he became Research Director of the Lactic Acid Bacteria and Mucosal Immunity Laboratory at the Institut Pasteur de Lille, France, specializing in fundamental research in the field of probiotics. During the same period, he started teaching food microbiology, hygiene, and ecology as a visiting professor at the Vrije University, Brussel, Belgium. In 2016, Bruno returned to Yakult Europe in Netherlands as Science Director Europe and continues his teaching at Brussel. Bruno is a member of the subcommittee on the taxonomy of Lactobacillus, Bifidobacterium, and related genera, and president of Pharmabiotic Research Institute. He is also a founding member of the ESLP and actively involved in activities of IPA Europe, ILSI Europe, and ISAPP relating to probiotics. He has more than 200 publications and more than 20,000 citations. We welcome you, sir, to this webinar. It's a great privilege for us to hear you. Thank you, sir.
Thank you so much for this very kind introduction, and thank you also all the participants for freeing some time to participate in this webinar. So it's really a privilege and an honor for me to be invited to give you this lecture on pro and prebiotics. Although I must say that the majority of my presentation will actually deal with probiotics, which is really my field of research, but I will definitely talk about prebiotics. So my first slide is actually showing also my potential conflict of interest, which have been already explained. So what I'm going to talk about is starting off with an introduction for the pre and the probiotic field. What are they? But before we go into, for instance, mechanisms, it would be really important that I explain a little bit about the activity domain of these pro and prebiotics, which is the microbiota. So my core of the presentation will be on some of the activity mechanisms of these pre and probiotics, so related to immune effects, microbiological effects, metabolic effects, with actually a few examples of a few clinical studies, which, despite all the research that has been done, are the only way to actually prove efficacy of either of the pro or the prebiotics. If there is time, I will spend some time on the regulatory hurdles and some economic benefits, but I think it's a lot already to talk about, but definitely I will end with some conclusions. So first, something about the definitions. So we are very fortunate in our situation because we have stable definitions of both pro and prebiotics for a number of years now, and they have been produced, at least for probiotics, already in 2002 by a committee composed by the Food and Agriculture Organizations and WHO, the World Health Organization. And the definition has been confirmed with some minor changes only in 2014 by the International Scientific Association of Pro and Prebiotics. The definition has three important parts in it. So probiotics are live microorganisms. They should be alive. They should be administered in adequate amounts, so the dose should be sufficient, and they should confer a health benefit on the host, which should be shown, as I already mentioned, by clinical studies. This is a very short definition, and it covers a lot of possibilities, as we will see later on. The prebiotic definition, of course, is different. It's not about live microorganisms, but prebiotics are actually chemical substrates that are selectively utilized by the host microorganisms, but again, with the purpose to have a health benefit. So if you would have a compound that promotes a pathogen, it definitely will not be considered a prebiotic. So the health aspect, again, proven through clinical trials, is really important. Maybe interesting to mention, if you make a product which combines the pro and the prebiotics together, then we talk about a symbiotic, which is the symbiotic nature of both the pro and the prebiotic. So a little bit of chemistry to understand what is mainly dealt with the prebiotics. So all of you know, of course, the glucose and the fructose molecules. If you combine them, you have sucrose. But if you extend further this chain with additional fructose molecules, then we talk about fructans. So the N2, 2 fructose molecules, delivers you the molecule Kestose, 3 will give you Nistose, etc. The interesting part is that this chain length can actually grow, and normally you could go up until the N value of 64. So 64 fructose residues linked to one glucose molecule. That type of molecule is called inulin. So it belongs to the dietary fibers category that all of you probably know, and it has a general name which is called the fructans. So this molecule is really present in many plants. It's estimated that about 36,000 plants actually make that compound, and they use it as a kind of storing of energy, just like starch is also a way to store energy in many plants. So what is important in the nomenclature we will use throughout is that if the length of the fructose residues is less than 10, we actually talk about oligofructose or fructooligosaccharides, shortened to FOS. So in the literature, you will find a lot of reference to FOS, but also to GOS. And GOS is actually the abbreviation of the same molecule in which the fructose has been replaced by galactose. So we're talking about galacto-oligosaccharides. If the chain length, however, is bigger than 10, then we call it inulin. And inulin is actually a mixture of different lengths of chains, which can be used in food and which can have some applications. For instance, if you look to the inulin, which is stored in chicory, which is a very important commercial source for the isolation of prebiotics, the average polymerization degree is 12. That could range between 2 to actually up to 24. So the application of this prebiotic in a technical context is also interesting, because the short length oligofructose is mostly used as a sweet replacer, while the long length inulin is mostly used as a fat replacer or a texture modifier, because it gives a better mouthfeel to the foods in which it is used. Medically, both are important, so clinically both have an important function, as because of the length, the chain length, they will not be digested in the small intestine. So as a sugar, they are not taken up, which has a benefit in terms of reducing blood glucose level, but they will be transported down to the colon, the large intestine, where they will be fermented by the microorganism presence. And that, of course, links up to the definition. It is a compound that is used by beneficial microorganisms, so they are fermented. Interesting to know is that the shorter chains, they will actually be fermented in the right part of the colon, while the longer chains, they will move further down the colon and be fermented in the left part of the colon. So by choosing the length, you actually have a possibility to work on where the fermentation will take place. And of course, if you have the mixture, you will have the possibility to have health benefits in the total length of the colon. What do they do there in the colon? So, well, I show you this picture of Professor Glenn Gibson. He's actually, together with Marcel Roberfroy, who described for the first time the prebiotics and the potential health effects. But both of these researchers have worked in the past, since 1995, really intensively on investigating the potential health benefits and the different structural ways of prebiotics. So what prebiotic do, they are of course considered part of the dietary fibers category. I will come back on that later in my talk. But initially they were said, well, they increase the good bacteria in our gut, mainly referring to bifidobacteria and lactobacilli. Nowadays, with modern metagenomics technologies, we know that it is much more than the bifidobacteria and the lactobacilli, which are actually touched by prebiotics. And there's in the literature reports of at least 100 other species that actually may interact with prebiotic fibers. So based on their degradation, they will produce beneficial metabolites. Very well known is the short-chain fatty acids like acetate, butyrate, propionate. And these, because they reduce the pH in the colon, they will actually also fight pathogens, because pathogens, as I will show later on, don't like very much acid environment. Because of the production of short-chain fatty acids, which is considered, especially butyrate, also an important energy source for the colonocytes, so the cells that line the colon, they will actually also affect the gut barrier and improve the gut barrier. Because of the gut barrier being implied, there will also be less bacteria that translocate maybe to the inner body. So we have also an indirect effect on the immune system and on a better defense, which has been measured by, for instance, a decrease in risk for allergy, because the barrier is more profound. There's less allergens that actually go through the intestine and come into contact with the immune system, giving rise to less allergy. And all the metabolic effects have also been described for prebiotics, mainly in increasing calcium absorption or decreasing protein fermentation, which is called putrefaction also. So what we see is there is a shift from the fermentation of carbohydrates by providing this probiotic, so that the effort and the energy sources are retrieved from carbohydrates much more than from protein, which is of course a health beneficial result. So most of you will actually know prebiotics maybe from another reason, and that is that the laxative effect is very well known. So if we look into that particular aspect, it's maybe nice to have a look at some figures. So in total, on a yearly basis, about 4 million Americans spend $500 million in buying laxatives. Although we know that food products like prunes and onions, they may actually have a similar effect because they actually contain these probiotic fibers in relatively large amounts. So studies say that, well, to be okay and to have the beneficial effect, the laxative effect, you would need about 30 grams per day optimally. If we look to the average consumption in North America, this is only 12 to 16 grams per day, while in Europe, a little bit higher, 22 grams. But none of these actually reach the 30 grams. So how is the situation in India? So I found this paper from 2015, which is actually looking into different population groups and making a difference between male and female for the consumption of dietary fiber in India. And so what is really striking for me is that there is a considerable difference between male and female. So if you check the paper, they don't explain this, but they say it depends on the food type. So maybe we can discuss this later on in the discussion, if there is really a difference in the food type between male and female, or why that difference is so clear in India. So let's move on to some of the mechanisms. And before doing so, I would like to explain a little bit more about the domain of the terrain of activities of pro and prebiotics, which of course is the human microbiota. So what is a microbiota? Well, in many cases, you will think about bacteria. But actually, it's important to know that the microbiota not only covers bacteria, but also yeasts and fungi, as well as viruses, phages, and parasites. And especially the phages, they may actually impact the levels of bacteria quite considerably. So it is really a very complicated ecosystem, which is called the microbiota, not only in the gut, but also in other places of the body, as you can see in this slide. So we have a microbiota not only in our gut, but also in the skin, the nose and the ears, the lungs, even the urinary and the genital tracts. They are all conquered by microorganisms, not only bacteria, but the list that I mentioned. And interesting, in this part of the slide, you can see that well, the composition of the microbiota in the different places is really different, depending on the substrates available, whether the atmosphere is aerobic or anaerobic. It all will, of course, influence the type of microorganisms that can grow there and survive. But very important for this presentation is the gut microbiota, so the gastrointestinal tract, which actually you can see even is variable depending on where we are. And this is very clear that we have this microbiota in our gut, because if something goes wrong with this microbiota, you will clearly see that some diseases or some problems can arise. And during our lifetime, like 33% of us will actually be confronted with some infectious bowel disease. 15% will be confronted with food intolerances, 10% with IBS or irritable bowel syndrome, 5% will be victim of antibiotic-associated diarrhea, 2% of food allergy, and then in minor percentages for other issues. So if you analyze the situation of the microbiota in these cases, what you will find that it is often linked to a so-called dysbiosis or a disbalance of the microbiota. So in life, we all know that there is a lot about keeping the right balance in many aspects of our life. But if we talk about a disbalance, then there must be a healthy situation also, of course. But the question is, how do we define a healthy microbiota? And if you ask different scientists, you will get different answers. And actually, nobody knows exactly how to define a healthy microbiota. And the reasons are very clear, actually. The microbiota, as already mentioned, is not only large numbers, but is also very complex. We have, each of us, like 200 to 300 different bacterial species. I'm not counting the fungi or the yeast in this case. So 200 to 300 different species, which are present in different concentrations and ratio. So if you see this figure here, and I don't want you to read everything, but it's just, you can see the distribution here is the relative abundance of 57 frequently encountered microbial genomes in a cohort of people. And what you see is that, well, not only there is a difference in concentrations for the different bacteria, but they also range quite substantially between the different people. And so one extreme example here is Streptococcus thermophilus, which, as you know, is one of the compounds of yogurt. So what this reflects is that people probably drink yogurt at this side of the graph, while people that don't drink yogurt actually have a lower level of them. So meaning that diet and environment will impact immediately in the composition of your microbiota. Is this microbiota more healthy than this one? Probably not. So there's so many factors that are important. So each microbiota, moreover, is unique, like a fingerprint. And just as like this, all of these fingerprints are functional, also different microbiotas can be extremely functional. So with different compositions of microorganisms in different ratios, the digestive system can work very well, and we can be very healthy related to all the functions that are governed by our microbiota. However, although it's quite difficult, there is a number of parameters that generally have been linked to an acceptable, as indicators, I would say, of a healthy microbiota. So first of all, there is the balance between the good and the bad bacteria. And this balance, of course, is in numbers. We should have more good bacteria than bad bacteria. But it's also in terms of metabolic activity, which is called, in the case of a stable system, a metabolic homeostasis. But also in terms of immune interactions and immune homeostasis. So these things can be measured in terms of laboratory tests, and will help you to be an indicator for a medical doctor, for instance, whether there is a dysbiosis or not. Also important for a healthy microbiota is sufficient diversity. There is some debate about this. For instance, very young children, they have large numbers of bifidobacteria. The diversity is rather low, and still they can be healthy. But later in life, diversity is generally accepted to be an important factor. Sufficient resilience. Resilience means that you have enough diversity to also react against changes. For instance, a diet change when you travel or when there is a stress period coming up, a resilient microbiota will be able to deal with that. And finally, there is a number of bacteria over the last years which have been described as being keystone species, a number of species that are thought to be critical in any one of us to be essential parts of the microbiota composition. What is for sure, however, is that we need those bacteria. There is this new concept which has been launched a couple of years ago, which is the holobiont concept, meaning that we with our bacteria, we are one. And actually we can find in nature proof of that we have evolved during millions of years together with our microbiota. If you look into simple organisms like this octopus here, you will see that it contains very little bacteria in this graph, and also the diversity is low. Well, if you go to the developed species like our human race, you will see that we have many more bacteria and that we have also a big diversity. So this is the result of this million years of evolution. And really, we should consider ourselves as being one with our microbiota. And this importance, of course, is linked to real functionality as well. So if we look to the microbiota of the gut, which I promised to be the focus of this talk, you can see that the number of bacteria will increase. In the stomach, of course, where the acidity is very high, you will have very few bacteria that can survive there. Well, if you go to the end of the gastrointestinal tract, to the colon, you will find very high numbers per milliliter or per gram. So these bacteria, they will contribute to the functioning of the gut. And the function of the gut is actually if you go to the literature, you will find three main topics. One of them is the digestion of food particles to compounds that can be taken up by the body. Bacteria can contribute by fermentation, for instance, or by adding enzymes that will help to degrade some difficult to degrade nutrients. Absorption is important. Of course, we need to take up the nutrients. So microorganisms, again, will help in making the compound small enough so that it can be easily absorbed. And there's also proof that they can help absorption of calcium, magnesium, and iron, actually. And finally, fermentation, which is happening in the colon mainly, where, for instance, the prebiotics that we talked about, or the dietary fibers we talked about, will be fermented to short-chain fatty acids like acetate, butyrate, which actually are important energy sources and also will be regulating the pH of the colonic fermentation. In this graph, however, I missed one big role of the microbiota, and that is protection. Protection by the production of short-chain fatty acids against potential pathogens. The production of hydrogen peroxide is also important, for instance, by lactic acid bacteria in the vagina. The production of bacteriocins, or by an interaction directly or indirectly with the host immune system. And in order to show the importance, I'd like to show you that slide. We should realize that in order to have this absorption function really good developed, we need in our intestine a very big surface. And actually, the surface of the small intestine is about the size of a tennis court. And this is achieved by, in the small intestine, the villi, and on top of the villi, on each cell, you have these microvilli. And these structures really increase the surface of the absorption surface in which nutrients need to be taken up by the body. And if you realize that during our lifetime, we eat like 65 tons of food and drinks, which is like the homologous of 10 elephants, you realize that defending this surface against pathogens, against toxins, is really, really important. And as such, we know that about 70% of the immune system is situated around our gut in order to defend that surface, a very delicate surface, a very big surface, to defend it against pathogens, I think, and toxins, for instance. So our probiotics will actually stimulate the immune system to a certain extent. I will come back in the next slide on how that goes, in order to fight potential pathogens. But it will also have an anti-inflammatory potential. Some of the strains will also have an anti-inflammatory potential, which could be interesting, for instance, in preventing or in fighting allergies, or also in preventing autoimmune diseases, where the immune system is actually reacting too actively. So how to explain this? This is an interesting paper from 2009, which simplifies in a really comprehensible way the different arms of the immune system in terms of fighting pathogens and in terms of dealing with beneficial bacteria. So if we take the pathogen arm, well, pathogens, they probably, or most of them, will have the potential to actually invade the intestinal epithelium after which they will be detected, of course, by the immune system. I explained to you that 70% of our immune system is located in that area. So they will be detected, they will be recognized as pathogens, and an inflammation response will be will be launched. And this inflammation response is mainly driven by a cytokine, which is called IL-12. I will come back. This is an important cytokine. I will come back on that in future slides. So this will drive the inflammatory response, which will destroy the pathogen, which is, of course, what we need. When the immune system, however, encounters a beneficial bacteria, something completely different happens. And of course, these beneficial bacteria will not be able to translocate, but the immune system is actually equipped to sense these beneficial bacteria in the lumen. So dendritic cells have the potential to penetrate to the lumen and sample intestinal content 24 hours a day in order to detect beneficial bacteria, as well as pathogens, of course. But when a beneficial bacterium is encountered, like a probiotic, you will not find an IL-12 interaction, but you will find these dendritic cells or macrophages to actually produce another cytokine, which is an important cytokine again, is IL-10. And that is a regulatory cytokine, an anti-inflammatory cytokine, which is able to stop the inflammatory response. If this would be an allergic response or an autoimmune response, IL-10 would be important in actually reducing the symptoms of those effects. So how do they actually work on this? So let's have a look at these immune effects. Later we'll look at the microbiological effects and also a few slides on metabolic effects. So one of the things I should start with, I think, in trying to convey a message of the complicated nature of what we are dealing with in showing a mechanistic approach for probiotics is that we know that probiotics seem to be doing everything. If you go through the literature, you can see that microbiota is involved, and therefore probiotics might be involved in obesity, in IBD, or the development, not the development, but inhibition of the development of obesity, may have links with IBD, liver disease, emerging infectious diseases, diabetes, atherosclerosis, metabolic syndrome. Lately, we have added a number of other problems like allergy, Parkinson's disease, Alzheimer's disease, cancer prevention, anxiety. So one would wonder, is it possible that probiotics can do all this? And in order to understand these different mechanisms, I think it's important that we should understand that it's not the strain itself necessarily that has all these effects, but it is its impact on the human microbiome, and it's the human microbiota quality which is actually linked to all of these diseases. So the effect of the probiotic is not directly with the disease, but is probably linked to an impact on the human microbiota, which through its multiple activities, I will come back on this later on, actually could have an impact on all of these diseases. But because of this complexity, of course, it is very, very difficult to show a particular effect. If I would be chasing a potential immune effect of a probiotic, I would definitely be confronted with potential metabolic effects as well. So this is shown in this slide. So we already discussed the immune stimulation and the anti-inflammatory response of immune effects. But there is definitely metabolic effects, which could be in vitamin production. We mentioned already short-chain fatty acids by the microbiota, for instance, by bifidobacteria, which are taken as a probiotic. The implication in bioconversions, also bifidobacteria interact very easily with other microbes in the colon. The fermentation with the production of butyrate and other short-chain fatty acids. There's microbiological effects. There's competition for places. There's competition for nutrients. There is the production of growth factors like vitamins, which will promote the growth of other beneficial bacteria. Or there is direct antagonistic molecules like peroxide, which will be active against potential pathogens. There is barrier effects stimulating the tight junctions between the epithelial cells, or the barrier function stimulating the mucus development, which is also protecting against pathogens. And finally, there's neurological effects like the gut-brain axis, which is very actually studied a lot nowadays. And finally, the endocrine implications. For instance, the impact on satiety hormones, which will tell you when you have eaten enough. Again, the complexity of the mechanisms which are involved. And what is very particular for probiotics is that you will be it's rarely possible to isolate one single mechanism. And the example I've shown here is, for instance, take diabetes. So diabetes, of course, is linked also to an immune. It's a consequence of a basic inflammation, physiological inflammation. So there's the immune system involved. It is, of course, diabetes also linked to the intake of food. So there might be endocrinological effects. There is barrier effects also because the glucose is going through. So fortifying the tight junctions might be important also. Diabetes and obesity is actually the same reasoning. There might be neurological effects. Mood could be involved. Metabolites like short-chain fatty acids, which reinforce the barrier function, may be implied. So you can immediately see that even if we would develop a probiotic for a particular application, we may actually count on a multiple mechanistic effect of these probiotics. What makes it additionally very difficult to study probiotics is that there is many external factors that will impact on the microbiota. Think about stress, lifestyle, smoking, for instance, the intake of medication, drugs like antibiotics, proton pump inhibitors, anti-acid medication. The genetics of the host, the age of the host might also impact. And of course, the diet, whether you take pro or prebiotics, whether you have sufficient dietary fibers or dietary microbes in your daily diet, will all impact on the probiotic or the prebiotic that you are taking. So first intermediate conclusion here. Well, it's obvious that probiotics have a huge potential because of their wide range of interactions with the host, whether this is directly or indirectly through the microbiota. However, we must realize that we are working with a very complex ecosystem, which is influenced not only by the signature of the microbiota, the personal microbiota, but also impacted by the environment, by diet, by stress, by drugs, which make it very difficult to predict the activity of a certain probiotic strain. Still, if we want to make health claims for a strain, we need to prove that effect. And this is the way to do it, of course, is through clinical studies. I will be giving a few examples later in the presentation. If we do these clinical studies, we should realize that there is a difference between a probiotic food, though, and a probiotic drug. This is important. A probiotic food needs to do clinical trials in a healthy population. That's not evident because what you are showing here is that you have a health effect in people that are already healthy. So how do you show that? I'll come back on that in an example. For a probiotic drug, the situation is maybe a little more simple because there you would use a diseased population to show a potential preventive or a curing effect, which is a completely different type of setting than you would have with a healthy population. In both cases, it might be interesting to understand the mechanism of action because that will allow you to screen in a lab, for instance, for strains that have even a better performance related to that mechanism of action. So let's have a look at the immune effect in a little bit in more detail. So there's many ways to select an optimal probiotic strains when you talk about the immune system. I've chosen to present this method because it's a very easy to understand, very clear way, and it shows very clearly, it's also reproducible. It shows very clearly how you can select different types of probiotic strains. So the source of the screening material is human blood, and you will need to take 5 to 8 donors. I will come back on this, why we need more than one donor to actually test. So you'll use the blood. This is the normal blood that is used in hospitals for patients that need blood transfusions. You will use that blood, put it into centrifugation with fecal, and you will sample the white blood cells, put them into contact with your bacteria that you want to test, incubate them in the right conditions, and these bacteria will induce in your blood cells some cytokines, the ones that I mentioned before, and which, I repeat again, are mainly the IL-10, as a model cytokine for anti-inflammatory potential, and the IL-12 cytokine as a model for the potential pro-inflammatory cytokines. But we could also look into interferon gamma or TNF-alpha, for instance. So what you see here is results for a number of different species and strains. And what you can see is that there is strains, in the case of the anti-inflammatory potential, like this Lactobacillus salivarius, which has a very high potential of inducing IL-10. So this would be a potential anti-inflammatory strain, while this model strain, which is a Lactobacillus acidophilus strain, doesn't have the same potential to induce to induce IL-10. But what you see if you look into the pro-inflammatory cytokines is actually what you could expect, mainly that a strain like the Lactobacillus salivarius, which has a large potential to induce IL-10, doesn't have the same potential to induce IL-12, which is actually the pro-inflammatory side of the immune reaction. But the strain which has a relatively low potential for IL-10 will actually have a high potential to induce IL-12. And if you look to pro-inflammatory cytokines as a whole, you will see that more or less you get the same profile with IL-12 and interferon gamma. So clearly this corresponds to the picture that we have shown before, in which you have potential lactobacilli that will impact this side, the beneficial side, with IL-10 as a main cytokine, or you will have bacteria that will promote the fight against pathogens by increasing the IL-12 ratio. So this is very important because it allows us, if you take an IL-10, IL-12 ratio, it allows you to actually see a difference between strains that have a potential anti-inflammatory effects and those strains which don't have that effect. And if you check this in animals, you will actually see, and this is a mice model for colitis induced by TNBS, you will see that strains with a high IL-10, IL-12 ratio, they have the potential to protect mice against colitis. While strains with this low IL-10, IL-12 ratio, they don't have that potential. So that is a way in the lab to actually select potential probiotics of interest. So whether you want probiotics which has an anti-inflammatory or probiotics that will help you fight pathogens. So clearly, it is important. Strain specificity is an issue, and you really have to be aware of this if you're studying probiotics. So not every probiotic will have the same capacities. Now, you remember that I mentioned you need 5 to 8 donors. So why is that? I think this slide clarifies this. So what you see here is donor 13, donor 14, donor 17, 18, 19, different donors. Each color represents a different strain. This is a cumulative plot, so you can see one strain is cumulatively plotted on top of the other. But what is interesting is that donor 13 in this case, and now we're looking into a plot for the cytokine induction potential IL-10, anti-inflammatory cytokine. Donor 13 has very little potential to actually produce that cytokine, while donor 18 has actually the opposite reaction. This donor is very suitable for the induction of IL-10. Which, interestingly, is confirmed if you look into the same donors, the same strains, the IL-12 donor. So the one which has a high potential for IL-10 is very low in IL-12. That's what you expect. And on the contrary, the one with a low IL-10 is very high in IL-12 potential. So that means that there is differences between the human beings also. So how we react to probiotic is also determined by our immune system. And so this observation may help to understand why, if you do a clinical study, you may have so-called responders and non-responders. People that don't respond to a probiotic strain because they are at that level. They will not respond to an IL-12 promoting strain. And this is interesting because in the lab where we did these tests in Pasteur Institute, we actually monitored patients, or it's not patients actually, it's healthy people which give the blood. We monitored those people over time. And what you can see is that their reaction will change over time. So in wintertime, when they have been confronted with viruses, their immune system may react in a different way than in summertime when they are perfectly healthy. So even within the time of a certain person, the reaction may be different according to the time you are trying out a probiotic. So immune system are very complicated, but there is tools that can help us to make decisions in the lab. Microbiological effects. So I'm not going to spend a lot of time in there because time flies. So this is the result of a number of clinical trials which have been done in children which receive antibiotic and as a consequence might develop diarrhea. So this is, I think the oldest study is here from 1990, so it's like 30 years of study work represented here. And what you can see is the general evaluation of the result. So if the line is at this side of the bar, then it is in favor of the probiotic. When the result is at this side of the bar, it is in favor of the placebo or the control. So what you can see here is that most of the studies, and there's only a few exceptions here, most of these studies will actually yield positive results. So they represent different types of strains, different conditions of use, different patient types. But if they receive antibiotics, these probiotic strains will support the children in developing less or fewer days of antibiotic-associated diarrhea. So how does this work? Well, this is again a relatively old slide, but I think it represents very well one of the possible mechanisms by which it could work. So this slide represents 3 pathogens that are able to cause diarrhea. So all of you are very familiar, I think, with Escherichia coli, with Campylobacter jejuni, and Salmonella enteritidis. It's actually the 3 main food pathogens all over the world. And so if you look to these 3 pathogens, they grow very well at neutral pH of 7. So you see the high curves and the 3 pathogens. But if you reduce the pH to 6, 5, 4, you can see that gradually the growth of the bacteria will be lower. And actually, they may even die off in the case of jejuni at a pH of 5. So that means that in vitro, in the lab, these pathogens are inhibited. So if you remember that a lot of the probiotics we talked about are lactic acid bacteria, so the production of lactic acid could be one of the mechanisms by which pathogens are actually eliminated. But there is, of course, more that is going on. And again, I like to use one of these papers, which is from a Dutch researcher in 2017 published, which shows for a number of known probiotic strains what could be the potential mechanism. So, of course, there is the production of organic acids, which is lowering the pH, but there's also competition for nutrients. There's bacteriocin production. There is for Bacteroides the type 6 secretion system mediated killing effect. There is competition for adherence or for places in the intestine. And finally, there's also an impact on the tight junctions, which is also an interesting effect that has been observed for a number of lactobacillus strains or lactobacillus species. So again, if you talk about probiotics, it's very likely that more than one mechanism will be active at the same time, which is also the case in fighting pathogens on the microbiological level. And then finally, an example of some metabolic activities. So this is a very interesting review that was just published August 2021, which is studying the effects and the mechanisms of probiotics, prebiotics, but also symbiotics, even postbiotics, in metabolic disease targeting the gut microbiota. And the general philosophy of the paper is that the gut microbiota can be influenced by these pro-pre-symbiotics in the production of bioactive molecules. And interesting is that they make a discrimination between bioactive molecules that will stay local in the lumen and can participate at the crosstalk processes. Examples of this could be short-chain fatty acids or bile metabolites. Or these bioactive molecules, they can translocate into the circulation and therefore have a metabolic and physiological role in the host. You can think about a limitation of lipid accumulation in adipocytes, for instance, in the case of obesity. Other mechanisms are that these bioactive molecules will be blocking harmful bacterial metabolites, for instance, LPS, lipopolysaccharides, which is cell wall component of some pathogens, gram-negative pathogens mainly, in order to be translocating into the host. And another mechanism is that these active molecules could interact with several key proteins involved in metabolic-related signaling pathways. And the PPAR gamma and the G protein-coupled receptors are just a few of those examples that are mentioned in this study. So most of these functions have been shown for probiotics, but will also be valid for prebiotics and for symbiotics as well. So we can study most of these mechanisms in the lab, but of course, as I said, the proof of the pudding is in the eating. And so what we need to do is perform the clinical trials. So for the prebiotics, this is an example of a number of studies that have been done with oligosaccharides, or the fructans, which I introduced, so the fructose oligosaccharides, as well as the galacto oligosaccharides. But you should know that, of course, there's other types of prebiotic-like molecules. So rather than fructose and galactose, you can have glucose-based oligosaccharides, xylose-based oligosaccharides, soybean-based oligosaccharides, etc. So all of these have been included in clinical studies, and the prebiotic effect has been measured. And it could be good, it could be moderate, good, or very good. And in this column, you can see what type of the microbiota, what bacteria, what part of the microbiota is actually positively influenced. Mainly it will be bifidobacteria and lactobacilli, or there will be a reduction, for instance, of the growth of Clostridia, which is also an important positive health effect. And here you can see the dose that has been used in these studies. But what I want to focus on, because in the story we've been talking about the immune system a lot, is how do you actually show in healthy population that your immune system is improved? Because people are already healthy. So that is a difficult question. And so if you set up a clinical trial, you actually have a need to seek for a population in which the immune system is not in maximum efficacy. It is still healthy, but it is not in an optimal shape. And these situations, for instance, could be people at old age. It could be people with chronic discomforts, like IBS, for instance, people with high psychological stress, or even physical stress. And what you can then show is that the probiotics can help to boost the immune system. So healthy people become less prone for further disease development as their immune system is being promoted. So let's give you a few examples. So this example is a study that was done in the UK by Professor Gleason in athletes. It's very well known that athletes during a very severe training program, their immune system will be less efficient. So what you can see is that the immune parameters go down, and they are more prone for infections, for instance, during winter time, for viral infections. So what Professor Gleason did, he included initially 84 athletes, which were very active, and therefore only 58 actually completed the study. 32 of them received the probiotic, and 26 received the placebo. So it was a randomized, double-blind, placebo-controlled trial, a high-quality study. The results of that, they got the probiotic at a dose of 13 billion CFU per day, and that for a period of 16 weeks. And the probiotic was L-Cassia. So the result was that there was not only less symptoms, but also less symptoms of upper respiratory tract infection. And both of these parameters were significantly changed in the probiotic group versus the placebo group, as you can see in this graph. But second and most interesting probably is the mechanism by which this change was, or this difference was explained. And the investigators, they actually observed an increase in the IgA in the saliva. So IgA concentration were significantly higher in the probiotic group than in the placebo group. And that was both at 8 weeks and 16 weeks. So again, indicating a possible mechanism by which the Lactobacillus casei was interacting, in this case, with the immune system. And this has been confirmed. This is all studies with the same probiotic strain, but done in elderly people. So I mentioned elderly people also have potentially a less efficient immune system. And what you can show, or what you can learn from these 3 studies, this is a Belgian study with elderly above 65 years, taking the probiotic for 6 months, is that the risk for infections going down as time goes on and the consumption of the probiotic goes down. This is a Japanese study with elderly above 83, average 83 years of old, 7 months of probiotic application, and you can see a reduction in the duration of the upper respiratory tract infections. And the last Japanese study, again with elderly, mean age 85 years, application for 6 months, in which the duration of the fever was monitored, and you can see also the probiotic compared to the placebo will increase as the time of the probiotic consumption goes on. A second example is for human papillomavirus. Again, a viral infection that needs to be fought exclusively by the immune system, because antibiotics will not work against viruses. So you have to have an active immune system in order to overcome viral infections. So this study was done in Belgium a couple of years ago. And in total, women were consuming, again, Lactobacillus casei for 6 months at a dose of 6.5 billion CFU per day. And what was monitored was the degree of infection with HPV, as well as the associated abnormalities, cellular abnormalities, which are considered to be precancerous. And so after 6 months, the results again clearly showed that there was a significant clearance, a significant better clearance of the cytological abnormalities in the probiotic group versus the placebo group. And also that HPV was cleared, the virus, the human papillomavirus, was cleared in 90% of the control patients only, while in almost 30% of the probiotic group, suggesting that the probiotic promoted the immune-mediated clearance of the virus and the related cytological abnormalities. So again, an indirect proof of the mechanism, the potential mechanism, which is the stimulation of natural killer cells, which are the ones that fight cells infected with viruses. So that brings me to the first set of conclusions. So the probiotic cascade effect. I think it's very important to realize that there is often not a single mechanism of action. So if you have a live microorganism and you take it in sufficient amounts, it might have a microbiological effect. The microbiological effect might have an effect on the barrier, on the metabolism. Barrier may influence how the immune system is reacting against it. And of course, a metabolic effect might have physiological results at the level of the host. And all of this may be reflected in endocrinological and neurological effects. But of course, the arrows can also be directly, so the microbes can have a direct interaction with the immune system or interact with the physiology of the host as well, or have endocrinological and neurological effects. So a really complicated scheme. If you are searching for a probiotic or you're looking for a probiotic for a particular application, you may need to consider all of these possible mechanisms of action. Another cascade is this nice pyramid, which was published also by the ISAP in 2014, which is also pointing out that if you look into probiotic efficacy, there might be different levels of specificity. So some of the probiotic properties, like the production of acid, lactic acid or short-chain fatty acids, might be widespread among many, many strains of probiotics. For instance, also the fact that they will compete for places in the intestine is a very common property for lactic acid bacteria, definitely for some specific probiotics. Then you have a second level of effects which are less obvious for a number of strains, but still quite frequent. For instance, like vitamin synthesis or an impact in the bile salt metabolism. And then you have the really specific properties, which we have been looking at in this presentation, namely like the immunological effects. And I can put up a complete similar story for endocrinological or neurological effects of specific strains, which have been tested and developed for this type of specific applications. So final conclusions. It's obvious that some bacteria have the capacity to positively influence the immune system, but we should be aware that this is rarely only one mechanism that is active at the same time. They will rarely only influence the immune system alone. For some strains, the immune activity has been clarified and has been linked to, for instance, cell wall components. For LGG, it's been shown that the proteins on the pili are important for the adhesion, which is the first start of the activity. But besides the immune activity, there's also metabolic and microbiological mechanisms, which are also investigated quite easily using in vitro models in the lab. We've shown also that the host status is important. And this is not only for the immune system, but also, of course, related to the metabolism and related to the microbiological effects of the probiotics. And finally, that the proof of the pudding is really in the eating. You need to show a probiotic effect through a clinical study. This is the only way to take into account the variation that is out there, the individual variation, the stress variation, the environmental variation, which is there, by selecting a sufficiently large group of people. You can actually show an average effect in the total population with this approach. So final conclusions for the prebiotics. Actually, I can simply say that most of the conclusions that I made for the probiotics will be valid for the prebiotics as well. because for the simple reason that prebiotics actually have an impact on the health beneficial microbes within the gut, which is, of course, those microbes that have similar effects or could have similar effects as the probiotic organisms you're taking. Probiotics are part of a dietary microbes category, food category. Prebiotics are part of a dietary fiber category. And while we cannot call every dietary fiber or dietary microbe a pro and a prebiotic because you have to show through a clinical study the health benefit, we really should realize that both categories, the fibers as well as the microbes in our food, have a really important role in maintaining health and in actually also preparing us for kind of challenges. So the resilience will be increased if we have a good quality of microbiota, which will be supported by both pre and probiotic intakes. And with this, I'd like to thank you for your attention during almost 1 hour. And if there is still any question, I will be happy to try and answer them. Thank you.
Thank you, Professor Bruno Pot, for a wonderful and highly informative talk on probiotics and prebiotics. The talk clearly puts into perspective the importance of probiotics, especially the gut microbiota. Indeed, gut health is one of the cornerstones of good health. But before we let you go, I have a few questions for you. Do you think the way we measure nutrition in food conventionally in terms of fats, vitamins, sugars, etc., is not the most effective way, and we need to be looking at food in terms of chemical composition of the microbes in the food, as well as the chemicals made by those microbes?
Yeah, so this is a very good question. So actually there is an increased interest in what I call the dietary microbes category. Officially, this category doesn't exist. But there is interest from ILC North America, which is now called IFNS. There's interest from IPA, International Probiotic Association Global. And there's interest from also the lactic acid bacteria industrial platform in trying to understand the health impact of live microbes in our daily diet. And very interesting, these organizations are working on publications which try to make an inventory of the consumption of live microbes in diets worldwide. And very interesting, you can see that India is one of the only countries where there is a systematic intake of live microorganisms. And it's also the only country we've found so far where in the guidelines specifically directed towards pregnant women, there is an indication to consume as much as possible live microbes. And so that is not by coincidence. So as I try to explain, the diversity is important in the microbiota, and by eating diverse fermented foods, you will add to the diversity. The diversity will take care of what you have said. The metabolites which are produced in the gut are really important, but not only within the gut, but also the metabolites which are being produced in the food might be equally important. And I'm talking about vitamin production. I'm talking about short-chain fatty acid production, which is extending the shelf life of the food also. So there is many reasons to consider this dietary microbes category. The problem is that legally this category is not recognized. So if you go to dietary guidelines worldwide, everybody will have, or most of the countries will have guidelines for the consumption of dietary fibers, but you will almost never find guidelines for the consumption of dietary microbes. And the purpose of the associations that I mentioned before is actually to try and create such a category, or to propose at least a category which is able to be added into dietary guidelines, so that a communication about the importance for as part of a healthy diet of this category is really there. And we hope that in the near future, there will be initiatives published that will go in that direction.
My next question is, as you mentioned, an important component of the human microbiome seems to be the human virome that is dominated by bacteriophages. Is there a difference between the bacterial-based probiotics and bacteriopatch-based probiotics?
Yeah. So that's also an interesting question. So nowadays, of course, there's a lot of information available on bacterial probiotics, but for the phages, there is very little information available. The reason for that is that phages are really almost strain-specific. So if you consider to use phages as a probiotic, you must almost know what strain is in the gut. in order to select the phage that will attack that strain. So in that case, I think it's going to be difficult. You would have to do, for instance, if you want to fight a salmonella infection with phage, you would have to first isolate the salmonella from which causes the problem, and then search in the lab for the right phages that would attack that particular strain of salmonella, so you can start the treatment afterwards. So in that case, I mean, there's a procedure that may take some time. So in that case, I think it might be interesting to start maybe early an antibiotic course in order to prevent people from dehydrating, for instance, especially if we're talking about children. This would be really important to act more quickly. So I think that the probiotic phages, that's a completely different story than probiotic bacteria, as I can see it now.
Yeah. Next question is, probiotics exert a great influence in modulating the immune system. So is there a risk of the potential immune system overstimulation in susceptible subjects, which can lead to autoimmune response and inflammation in case of a colonization with a relatively higher load of bacteria?
Yeah. Well, this is also a good question. It's a kind of difficult question. The practice shows that that is almost never the case. So because we are working with a complex ecosystem, if you would take too much of the probiotic, it will not be able to establish within the complex ecosystem at these high levels. So the probiotic will also be eliminated by the rest of the microbiota. It's not only the probiotic that influence the bad bacteria, but the bacteria in the gut will also influence the probiotic. So first of all, you will have a reduction at the level of the stomach. That is one point. And second, you will have, of course, the competition with the rest of the ecosystem. So the risk of having an overgrowth of your probiotic is really very low. The only thing that has been observed is in some cases that the probiotic could translocate into the bloodstream. And there it could be the cause. This has been observed for fungi, for yeasts mainly. But then also that yeast is, as it has a probiotic nature, it's mostly eliminated without further consequences. So there's been very few reports in the literature about infections that have been caused by probiotics. So in order to illustrate this, I can say that maybe also that there is a lot of tests that have been done with probiotics in exactly very frail patients. So patients with HIV, very old people which have a very weak immune system have been treated beneficially with probiotics, which shows that really if you have the right probiotic strain, then the risk for infection, the risk for negative effects is low. But again, it cannot be excluded, and it's always up to the medical doctors that prescribe the probiotics to decide whether this patient is able to take a particular strain of a probiotic.
Due to shortage of the time, we cannot actually take up all the questions. I'll just take one question from Sunil Shrivastavaji. He's requested to ask the question himself. So please go ahead, Sunil Shrivastavaji.
Yeah, excellent lecture, Professor Bruno.
Thank you.
I know one Peter Poy, who was the director of the London School of Hygiene and Tropical Medicine. He is one of my heroes. But today I've discovered another hero. I listened about you much, but I never had a chance to listen to you. So my question is, diabetes is a very complex disease. And you talk of microbiota, and I think no part of the body, system of the body is not affected by diabetes or has an influence: endocrine system, metabolic, obesity, stress, neurological, immunological, inflammation, whatever. So have you worked on any microbe that you can say, well, this can, or you might have in your clinical trials, reverse the diabetes by using that? So that would be a wonderful recipe for curing diabetes. So your comments on that, sir. Do you have some?
Yeah, unfortunately, I didn't work on diabetes. It is, as you said, a very complicated disease. So you have the genetic variant, which probably will never be treatable with probiotics. But then you have the metabolic variant. And there's quite a number of studies that actually show that if you can improve the gut barrier, and this is definitely some probiotics have been developed to do that, you actually will reduce the inflammation, the physiological inflammation, which is linked to diabetes. So, and actually, there is studies that have shown that you can reverse the situation. Of course, it's not probiotic alone. It needs also diet adaptations, because in many cases, this type of diabetes is also linked to overweight or even obesity. And so if you can reduce the inflammation as a whole in the adipocytes, so treating also the obesity effect, and the diabetes will also automatically be reduced because the inflammation will go down. And I think the only literature that I know is really in that aspect, trying to reduce inflammation by choosing anti-inflammatory probiotics, which also has an effect on the barrier in order to reduce the risk for the inflammation as well. So there's definitely a lot of research in that area.
So since you are linked with Yakult in a way, I just want to know, in India, we have a very high sugary content in the Yakult. I do not know about the European specifications or the guidelines. And diabetes is a major problem in this country. So can we have some other substitute for the sugar, for lactobacilli, casei to survive and multiply and whatever it is?
Well, that's a good question. So first of all, I'm not really aware of the situation in India. So in Europe, we have the same product, probably, but we also have a light variant in which the sugar is down. So we call it Yakult Light, and also Yakult Plus, in which the sugar is considerably down. So that is probably one of the solutions. And I'm not aware if this product is in India, actually. But to complete the answer, so the sugar content in the Yakult Original, that's probably what you refer to, is indeed rather high, but the volume is 65 ml, is relatively small. So the sugar which is contained in one bottle of Yakult as a whole is not that high, and is actually able to match with a daily sugar intake, a normal daily sugar intake. So if you have a dietary advice, you can declare this amount of sugar for the product, and it will help to limit the number of the amount of sugar to the daily recommended intake for the type of person you are. I mean, men probably need more sugar than women, so you need dietary advice, and Yakult could be perfectly part of that dietary advice. The active compound, of course, in the product is the LCS strain, and that is a different discussion. This is not linked to the sugar.
Okay, thank you very much. Merci beaucoup.
Thank you. Yeah. Thank you, Professor Bruno, for answering all the questions so patiently. Honestly, we would love to carry on asking you questions and to draw on your past experience in this field, but time permits us to go only as far. The knowledge that you have shared with us will help us immensely in improving our understanding of probiotics. Thank you once again. Now, I will request Dr. Aarti Nigam, Head of the Department of Microbiology, Institute of Home Economics, to deliver the vote of thanks. Dr. Aarti Nigam.
On behalf of entire faculty of Institute of Home Economics, I, Dr. Aarti Nigam, offer my sincere thanks to Professor Bruno Pot for accepting our invitation and delivering such an informative talk. I am sure all the participants are benefited from your talk, sir. We are hopeful that in near future also you would be able to spare some time for updating our students on probiotics. Thank you so much, sir. I also express my gratitude to MSI President Professor Deshmukh, Delhi President A. Archana, and State Coordinator Dr. Sunita Agarwal for organizing this, for participation and organizing this event. I am also thankful to Dr. Pratishtha for coordinating this program. And lastly, I am thankful to all the participants. Thank you all.
Thank you, ma'am. Before closing today's session, I would request